Hi students, welcome to Lycus Academy. In this section, we are discussing about basics of transcription. Before entering into transcription in detail, we must have an intense knowledge in basics of transcription. Transcription is the process of synthesis of RNA from DNA. Transcription is the first stage in gene expression. The genetic information stores in DNA. This information flows from DNA to protein. This flow of information takes place in a sequential of process, transcription and translation. During transcription, the genetic information in DNA transcribed into mRNA. Then during translations, this mRNA translated into protein. This protein involved in further process. As we all know that DNA has a double-stranded structure. One of the strands acts as a coding strand and the other one is act as a template strand. Coding strand, otherwise known as sense strand or non-template strand. It codes for the genetic information and it represents in 5', 3' direction. RNA is identical in sequence with this coding strand. The other strand, template strand or non-coding strand or non-sense strand, acts as a template for RNA synthesis. It is complementary to coding strand and represent in 3', 5' direction. In this picture represents the coding strand versus template strand. In this, the red colored strand that is in the direction of 5', 3' represents coding strand, and the blue colored strand in the direction of 3', 5' represents the non-coding strand or template strand. This non-coding strand has complementary sequence to that of coding strand, so it acts as template during RNA synthesis. RNA synthesis occurs with the help of RNA polymerase. RNA polymerase is the main enzyme involved in transcription. This enzyme acts as a catalyst for RNA synthesis. It uses single-stranded DNA to synthesize a complementary RNA strand. RNA polymerase binds to promoter and initiate transcription. Promoter is the sequence within the DNA, where transcription starts when RNA polymerase binds to the promoter. Start point is the base pair within the promoter. Promoter usually surrounds the first base pair that is transcribed into RNA. This is known as start point where transcription starts. From this point, RNA polymerase moves along template and synthesizing RNA until it reaches the terminator sequence. Terminator is the sequence within the DNA where end of transcription occurs. When RNA polymerase moves along the DNA, when it reaches the terminator sequences, the transcription stops. RNA polymerase then releases the DNA template. Then this transcription unit is the sequence of DNA extending from promoter to terminator. It consists of a promoter followed by coding genes, then finally the terminator. Transcription unit may include one or more than one genes, and RNA synthesis occurs in 5', 3' direction. Then comes to the term upstream sequence. Upstream sequences prior to the start point, it represented by minus values, that is minus 10 element, minus 35 elements are examples of these upstream sequences. Then downstream sequences are the sequences that occurs after start point. Transcription proceeds from upstream to downstream. Then comes to the term primary transcript. Primary transcript is the immediate product of transcription before any modification. It is known as pre-mRNA. It is the representation of the DNA structure. There are two strands, that is template strand and non-template strand. There is a region promoter where RNA polymerase binds and initiates the transcription. Transcription start site is the site where first base pair added. It is followed by RNA coding region, then the terminator where transcription terminates. The sequences prior to the transcription start site is known as upstream, and the sequences after the transcription start site is known as the downstream. Then regulatory proteins. Regulatory proteins are the proteins which determine whether a particular gene is available to be transcribed by RNA polymerase. For example, activators, silencers, suppressors, etc. Then talk about the transcription bubble. RNA transcription takes place within the transcription bubble. During transcription, DNA is transiently separated into single-stranded, and the template strand used to direct the synthesis of RNA strand. And the length of transcription bubble is 12 to 14 base pair. RNA polymerase creates a transcription bubble when it binds to the promoter. RNA polymerase moves along DNA, bubbles also moves along it, and RNA chains grows longer. This represents the transcription bubble formation. During transcription, the enzyme RNA polymerase binds to DNA and it melts the DNA. So the DNA temporarily separated into two strands. It leads to the formation of transcription bubble. Finally, this will give you a clear picture about the flow of genetic information from DNA to protein. In DNA, there is coding strand as well as the template strand. Both have complementary sequence to each other. Coding strand code for the genetic information and the non-coding strand act as template for RNA synthesis. Therefore, during transcription, the mRNA transcript form. The mRNA transcript uses non-coding strand as template. Therefore, the mRNA sequence is complementary to that of the sequences in template strand. But the mRNA sequences are similar to that of the sequences in coding strand. So there is a copying of genetic information from the DNA into mRNA. This mRNA further processed into protein with the help of the process called translation. During translation, each codons in the mRNA code for a particular amino acid. These amino acids are combined together to form a polypeptide, and it will forms the protein. Hope this video useful to you. Stay tuned to next video. If you like this video, please share, like and subscribe like Gaze Academy, your educational catalyst.